I'm trying to give you an overview of the human studies that have been conducted and how to interpret what has been done so far, and more importantly, what's not been done so far, what we should be striving to do. Before anyone wants to take microbiome seriously, this is the stuff that you get from your patients. So you have to actually deal with the excrement. Remember, it sounds all very glamorous right now, but when the fume starts wafting, you will figure it out. This is, I need you to remember this, okay? So, what is the clinical evidence of microbial change in hepatic encephalopathy and cirrhosis? Why do we think this may actually be an issue? We have known for a very long time that bacterial translocation, either by actual mesenteric DNA bacterial translocation, blood DNA, or DNA in the ascitic fluid, is a harbinger of negative outcomes in cirrhosis. And many elegant studies, including by some of our audience members, including Dr. Albilos, have said that. And treatment with non-absorbable antibiotics can treat hepatic encephalopathy, reduce bacterial translocation, and improve outcomes. And treatment with laxatives and probiotics, including capsules and foods, can improve outcomes in hepatic encephalopathy. And these are patients who are comatose. You give them a laxative or whatever, and they wake up. So you know something is happening. The fortunate thing now is to be in a situation to find out why it's happening, not if it's happening. So these are the things that we find out in brain-gut interactions. This is, as you see, there's one organ missing here. So it was written by a gastroenterologist. So I've inserted the liver here. Thank you, Dr. Forbes and the like. So food-derived substrates and neurochemicals are actually define the microbiota. Not only do they define the microbiota actually respond to them. They can either go via the enteric nervous system directly to the brain, or are absorbed by the portal circulation. And here I'm inserting the liver, because if your liver is diseased, this has a major role to play in what happens beyond this. So liver-brain is a separate issue, and microbiota and the portal circulation in the liver is a separate issue. The hypothesis then being that the brain is then affected. It then affects the brain behavior and cognition, and this food cycle starts. So if you're depressed, anxious, chances are the microbiota make you more depressed and anxious, you eat more, and the vicious circle continues. This is not as simplistic as what it is, because you are what you eat, okay? So if you're eating a cheeseburger, you're fat because you ate the cheeseburger, not because your microbiota made you fat, okay? So the liver, let's talk about a model for hepatic encephalopathy. Poor synthetic function, decrease in albumin, and decrease in bile acid synthesis and secretion, and increase in portosystemic shunting leads to dysbiosis, overgrowth of bacteria, altered ratios of autochthonous, which are the beneficial bacteria, to the potentially pathogenic ones. And more importantly, it's the altered function and not just the composition. This can lead to an impaired intestinal barrier with the reduced bile acids and altered autonomic function. If you remember, the enteric nervous system is a critical component of the brain-gut interaction. This can end result be a systemic inflammation increase in endotoxemia, and increase in gut bacterial products. That can potentially result in overt and minimal hepatic encephalopathy. Of course, this is not discounting the fact that certain toxins that can cause liver disease can also directly affect the brain, such as hepatitis C and alcohol. So like the session beforehand, this has to be a sum total of what all happens with the patient. Normal gut microbiota at a phylum level, for those who are not familiar with microbiology as I was, the typical stool microbiota is divided into these phyla. The majority of the phyla are between Firmicutes, which are the gram-positives, and the Bacteroidetes, which are gram-negatives. The phyla of interest to us usually is Proteobacteria, which includes almost every single gram-negative pathogen that you can think about, especially those that are important in cirrhosis, such as E. coli, Klebsiella, Pseudomonas, Pasteurella, etc. The rest I've not even labeled because they're not that specifically important in the studying of the composition of the stool. The composition of every other niche is a little bit different, and especially in the saliva, etc., you have much more difference in the composition. So the hierarchical organization of bacteria goes from phylum. So this is the calling card for E. coli. Okay, it goes from class, order, family, genus, and then finally species. Beyond species are strains. So you see, this is just one organism right here, has multiple strains. So the mind fairly boggles when it comes down to how many strains we might be carrying inside us and what they all are doing in concert. So the thing to remember, most of my talks are going to be at the family level or the phylum level or the genus level. So anything you have A, C, E, and an italic means it's a family. So the levels of study of microbiome research are tremendously complex. The first and the most simplistic things are which microbes are present in the gut. This could be in the stool, GI tract mucosa, or in peripheral systems. The prior studies were culture dependent, so it's like if you have a hammer, everything is a nail. So if you're looking for a specific bug, you will find it, but that doesn't mean that the other bugs do not exist. The culture independent techniques, which are a little more DNA sensitive techniques, actually look at the highly conserved V2 region of the 16S RNA, which is prokaryotic. So this does not differentiate between a live bacterium and a dead bacterium, but it does tell you what bacterium is present without a shred of a doubt, or at least was present in a little while ago. To me, the more important things are what are the GI microbes doing. Metatranscriptomics are the mRNAs, the proteomics is the protein, and the metabolomics are the metabolites that you think can be attributed to potentially an interaction between the gut microbiome and the host. And the last thing is what microbial genes are present in the GI tract in the metagenomics. And ultimately, you have to remember that if this thing doesn't affect your patient, it really becomes of an academic significance. You have to try and link it to something which is potentially important for your patients and whatever model you're studying. So the outputs are type of bacteria, presence or absence, richness, diversity, and relative abundance. Relative abundance is a little tricky to understand in the sense that you may have a total bacterial count of say 100. And if 50% of them are firmicutes, it's a 50% abundance. However, if the bacterial count is 10 million, 50% is still a lot of bacteria. But 50% in relative abundance means just the proportion of what is found. It has no bearing on the total number of bacteria that are actually present in the system. So the specific issues for us to interpret and design studies in HE and microbiome. Number one, are the microbiota changes simply due to advancing liver disease? Patients with HE typically have advanced liver disease. Second, are there specific microbiota changes in the mucosa, in the stool, or both in patients with HE? What are the effects of HE treatments and how much can they confound the actual treatment of the microbiota? Is it the relative change in microbiota abundance or a change in function that results in overall improvement or poorer prognosis, as the time might be? And are these bacteria just markers of a poor prognosis, or do they actually have pathogenetic roles to play in disease progression? So let's talk about the effect of advancing liver disease and changes in stool and mucosa. This is a principal component analysis, and you'll be seeing a lot of these. Forget your t-tests, forget your Kruskal-Wallis, forget everything that you've learned about regular statistics, and Dr. Marchesi is going to go over this, I'm hoping, in a lot more detail. Further, if they're clustered together, you're not, okay, well, then we might as well. Okay, so the ones with the open squares are controls. They are all clustered together. One dot is one person's entire variability of their microbiome, which means these vary together, whereas the serotics are clearly apart. This is one study done by Chen et al. This is a study done by our group, initial study. You saw the cirrhosis no, which are controls, are in blue. Cirrhosis yes are in the red, and they are far apart from the ones who are controls. This is a latest paper from Nature, which again differentiates healthy controls right here and cirrhotic cases right here. So there is sufficient data now to figure out that patients with cirrhosis in their stool are different. What about the specific bacteria that we are talking about? I like to talk about these three families as the three musketeers because every time they are present means something good is happening. So these are the autochthonous bacteria, the Clostridium cluster 14, Lactospiraceae and Ruminococcaceae. Don't ask me what they do, but they are markers of something good. They are markers of every time you look at any study, even Crohn's disease, ulcerative colitis, infectious disease, if these are overrepresented in healthy controls, more often they're not, and their absence is actually a marker of something bad happening. So these are much more in controls than they are in cirrhotics. And this, the Child-Tokart Pew score and Lactospiraceae. You see, as the Child-Tokart Pew score advances, your Lactospiraceae relative abundance goes down. Whereas Streptococcaceae, I don't know if you can see, but I can say it, Streptococcaceae goes up. So it does vary with the amount of liver disease. This is the Nature paper that everyone keeps on talking about. This is their phylum change. And they divided patients with cirrhosis. No way. 5 minutes left? Damn it, okay. Okay, basically they didn't account for changes in their patients with decompensated and compensated. Majority of their changes were because of decompensated cirrhosis. Same thing here, now I'm running through. Okay, blah, blah, blah. Stool microbiome, different. So this is the dysbiosis ratio, good stuff on top, bad stuff in the bottom. So it tells you it's a ratio of how bad things appear in our patients. And from healthy controls to compensated to decompensated to inpatients, it goes down. Microbiome in cirrhosis remains stable over time. If you take patients in microbiome, patients' stool cirrhosis, and you check them over 6 months, the microbiome remains relatively stable, so it can be used as a biomarker. What about patients who are nearing ACLF or death or in the hospital? These are MELD-matched patients with cirrhosis, so the liver severity is taken out. Anything in the red is controls, anything in the purple are patients without an infection but are in the hospital, and anything that are infected are far away. You can see despite having this extreme form of inpatient issues, these patients are still clustered with the controls, which means that this is highly conserved even in this patient. So if you're infected, you're much more likely to have poorer prognosis. What about the surface? The surface has different changes in the microbiota of the similar changes. I'm sorry, let's go over. Lactnospiraceae, this is the genera. There are similar changes in the microbiome of the sigmoid mucosa in patients with cirrhosis, as you would find in patients in their stool as well. And the sigmoid mucosa, again, same thing, healthy controls and patients without encephalopathy cluster together, patients with encephalopathy cluster far apart in patients with cirrhosis. What about it being a compositional or the functional change? The seminal point in this Nature paper was they actually studied the function of the bacteria. And they studied what genes were activated. Did they tell you what these bacteria were actually doing? This is too complex for me to explain in those 2.5 minutes that I have remaining. But it tells you that the function of the bacteria is also different. The genes that were activated were actually different. But does that mean that the metabolites produced by this were measured? No, the metabolites were not measured. So you have to have a whole pathway from gene activation to RNA to protein and the metabolite for something to be actually attributed to a microbiome. And they compared it to diabetes. I'm going to go forward. Dr. Filippo's group actually went one step forward, and they found a lot of serum metabolomic changes, but I'm going to concentrate on trimethylamine oxide, which is a co-metabolite between the diet, the microbiome, and the liver. And that is important because in patients with MHE, it was changed. Fecal metabolomics in cirrhosis have also shown that the aromatic amino acids, which can produce a lot of false transmitters, are much higher in patients with cirrhosis compared to controls. And the other function, which is bile acid dehydroxylation, is also significantly impaired in patients with advanced liver disease. So there's a group of highly conserved bacteria, which include the autochthonous bacteria, that convert primary bile acids into secondary bile acids. So that can also be used as a functional marker of the microbiome's health. And as cirrhosis advances, this conversion reduces because we know that these bacteria disappear in these patients. HE treatment on microbiota, you're making me run very fast. Okay. Long cliff notes. Lactulose does not have an effect on the microbiota composition. Does that mean it doesn't work? No. It probably has an effect on microbiota function. In healthy patients who were given lactulose, there was no change in bifidobacteria, no change in lactobacillus, contradicting prior studies which were culture-based. And this is because the lactobacillus and bifidobacteria may have changed from 0.01% to 0.1%. And that may have been enough to cause a beneficial effect to the host. But that doesn't mean it shows up in the 100% relative abundance. In cirrhotic patients with prior overt HE, when you withdrew lactulose, there was minor changes in the microbiota. In patients who had before and after the first HE episodes, there was hardly any change in the microbiota then. And lactulose withdrawal, if you remember TMAO, this is a study we had done in concert with collaboration with Simon, that the TMAO was reduced in people who ultimately recurred with hepatic encephalopathy. Rifaximin, again, no significant change in the microbiota beyond what you would expect in patients with liver disease. In the US, rifaximin and lactulose are much more used. Rifaximin is only given to people who are much more advanced. So these people had a worse microbiota compared to people who were not on them. And same thing, the rifaximin changed the bacterial associations. If I start explaining this side, we'll be here till tomorrow. But what it means is after rifaximin, the associations with the bacteria and the metabolites in the serum and the urine changed dramatically. Rifaximin reduced the fecal secondary bile acids, which it means it was doing something to the bacteria. What about probiotics? This is a study that's going to come out with Dr. Dhiman. They do a lot of good in the patients. But how do they do it? Unlike rifaximin, probiotics did seem to actually change the microbiota composition. In addition to that, they changed the metabolomics in the serum and the urine. And most of the metabolomics were actually related to vitamin C and vitamin B metabolism, which these patients were also given at the same time. And there is studies saying that the vitamin C can impact the intestinal barrier in these patients. So this may be a reason why these work. What about function and outcomes? I'm not going to go into details, but bacteria are associated with cognitive function. They are associated with function of the systemic inflammation in patients with HE. They are associated with inflammation and ammonia, including strep salivarius in this earlier Chinese study. And this is our unpublished data right now. It actually was a poster at EASL in which we compared magnetic resonance spectroscopy with the stool microbiota in patients with cirrhosis. As you're aware, if you have high glutamate and glutamine, you're much more likely to have poorer cognitive function and worse encephalopathy. And the bacteria, high glutamate was correlated in all three areas, white matter, gray matter, and anterior cingulate, with MELD, ammonia, and negatively correlated with the good bugs that we were talking about, lacno, rumino, and Clostridia. And it was the reverse in myoinositol. So high glutamate and low is compensated by low myoinositol and choline. And the reverse happened when this was done. Does that mean that the lacnospira actually went and reduced the myoinositol, reduced the glutamate? No. It just means that the same people have same things going on at the same time. And it is a functional marker of something happening in the brain. What about infected cirrhotic patients? Stool microbiota collected within 48 hours, which points, and we defined organ failures as was defined by the Cliff Consortium and by our thing. And we used death and organ failures as an outcome. These are healthy controls right here. The middle band are people who did not have organ failures in 30 days. And these are people who had organ failures in 30 days. And this was related to higher endotoxin and lower lacnospiracy and veillonelacy. What about death in 30 days of patients admitted to the hospital? Normals right here, non-dead right here, and the dead people up there. So this can actually predict what your patient is going to do in the next 30 days if they're actually admitted on the day one. So in summary, thank God. Cirrhosis, you made me rush through a whole thing. Okay. Cirrhosis is associated with dysbiosis in stool and sigmoid colonic mucosa that is associated with the severity of the underlying disease. HE treatment can cause changes in the microbiota composition, but functional changes are much more likely. And as we talked about, MHE therapies may affect microbiota differently. The probiotics may actually change the microbiota, whereas things like rifaximin may actually change the function of the microbiota. Microbiota changes have been linked to patient function and outcomes in cirrhosis. I would like to thank you all for your attention and bearing with me.